To your second lecture on chemistry, water, and organic polymers. The first thing we're going to look at today is chemistry, and we're doing a little bit of chemistry in biology in order to get a little bit of background so that we can look at things and understand nutrition and exercise science for this class. These are the common misconceptions for bonds. Bond formation does not influence the charge of atoms and molecules, meaning atoms and molecules have inherent charges, and they're not influenced by surrounding atoms or bonds they may form. So think about that as you're working through this module and see if you're able to disprove it at the end. All right, so we're going to look at some different types of bonds, and the first type of bond is a covalent bond. Covalent bonds are really strong bonds. They dominate organic systems. And when you have a covalent bond, there can be two types. They can be polar or they can be nonpolar. Bonds that are nonpolar have an equal sharing of electrons. And so what you can see in this image is these guys coming closer to one another, the atoms, the electrons actually moving around both of those atoms. And it's a really strong bond. And this would be a nonpolar covalent bond because it's an equal sharing of electrons. We can contrast that with polar covalent bonds. Polar covalent bonding is an unequal sharing of electrons, and water is a great example of this. First, let's define polar. If something is polar, it means that it has some sort of charge on it. It could be a partial charge, or it could be a full-blown charge. And the reason we want to understand whether something is polar or nonpolar is to understand whether it's soluble in water. So if something is soluble in water, it's polar, and it just means that water can form points of attachment, bonds. It's attracted and it can surround that molecule. And so if we look at water itself is polar. And the reason it's polar is because there's a really big proton difference between the oxygen atom and the hydrogen atom. So what ends up happening is because oxygen has more protons, and that's positive, right? That means that the electron, the one electron that hydrogen has, ends up spending a lot more time around the oxygen atom. So it's an unequal sharing of electrons. And when an electron hangs out more often over here, that negative charge brings on an actual partial negative charge to that oxygen. We're going to be dealing with two what we call electronegative elements in this class. Those are oxygen and nitrogen, and both of them do this. They engage in a polar covalent bond anytime they are bonded to hydrogen. So here you have an O to H bond, or this could be an N to H bond, and both of them would be polar covalent. All right, so you have a partial charges going on here. You can see that you've got partial positive charges on your hydrogen, and the reason is because it's losing that negative electron a lot of the time. So all that's left here, most of the time, is the positive proton. So this gets a partial positive. You can think of this as oxygen and nitrogen as being greedy elements. They are greedy for electrons because of that strong proton difference. And so they hoard electrons. Electrons are negative, so they take on a partial negative charge. All right. So what this does is it makes water polar. And other molecules that have O to H or N to H on them are going to be polar as well. And the reason is because they have some partial charges. We defined polarity as having either a partial or a full-blown charge. So not surprisingly, water molecules like other water molecules, meaning they can come in and bond with each other. And we're going to talk about that type of bond in just a moment. So you have some terms down here, and all of them are synonymous with one another, and it's just a good thing to keep in your terminology tool belt is to know the term polar, which means there's a charge, any type of charge, or hydrophilic or soluble. All of those are the same. There's a charge and they like water. So the term electronegativity pertains to, in particular, oxygen and nitrogen. Those are both going to be the electronegative elements that we're looking at. So when you look at molecules, you're going to be scanning them for O to H bonds or N to H bonds. Electronegative just means that they are greedy with electrons, like we described in water. The other cause of polarity is ionic bonds in ions. And so I'm going to show you the difference between those two in a video. Ionic bonds are different than covalent bonds because that electron from Na is completely given up. Covalent bonds don't ever completely give up their electrons. They just share them either equally or unequally. So that's an ionic bond, and it brings on a full-blown charge. So water definitely likes ions. Nonpolar covalent bonds are equal sharing of electrons. So you'll notice that those little yellow guys, the electrons, are being equally shared between both atoms. That's not the case for polar covalent. So here's oxygen, which is electronegative, and it hoards hydrogen's electron most of the time. Not all the time, but most of the time. And that brings on a slight negative charge for the oxygen and a slight positive charge for the hydrogen. So there's your slight negative and your slight positive. Hydrogen's just showing its proton. On the flip side, something that is non-polar covalent is going to have that equal sharing of electrons like you just saw in the video, but I wanted to point out some examples that you see of this in real life. Non-polar are things like your oils and your butter. You can also note anything that has a waxy consistency, like the wax on the coating of a leaf. All of those don't like water. Water really shies away from those. All right, so we've talked about covalent and ionic, and now we're going to talk about hydrogen bonds. Hydrogen bonds occur between two polar covalently bonded molecules. Okay? So we just talked about water as having polar covalent bonds. So if water gets around other water molecules or anything else that has O to H or N to H, it forms what's called a hydrogen bond. These are really weak, but of tremendous biological significance. You're going to have these in proteins, you're going to have these in DNA. These are really, really important bonds, and their importance lies in their weakness, which is interesting. So this weak association, we designate that as a dotted line. And you can see, in chemistry, opposites attract. And so you have the plus side of a hydrogen, that partial plus, being attracted to the partial minus of the water molecule next to it. And this is a very dynamic bond. Water molecules at room temperature, so it's a beaker of water, those water molecules will be breaking and forming every few trillionth of a second at room temperature. Okay, that's not the case for ionic and covalent bonds. Those are strong bonds. Hydrogen bonds are very weak bonds. So why do we care about polarity? What's important here? What we're really concerned about is, you know, in living systems, what types of molecules can easily move through water, which is mostly what the cytoplasm is, and what types of molecules have trouble. If you have a molecule that likes water, what water molecules do is they form something called the sphere of hydration. And so here you have a salt cube, and that salt cube, if you drop that in water and you don't stir at all, if there are enough water molecules, eventually water will surround each individual atom. And these are ions in this case. You see those full-blown charges, so obviously water loves this. So the importance of polarity, what we want to understand is what can be soluble and what is not soluble. Your common misconception for water is that all living things need water, but water has no influence over abiotic systems like climate. So this is saying that water is only necessary for living things. It has no influence over abiotic factors. Okay, this next section is going to talk about water a little bit deeper. We want to learn about the properties of water, and so I'm going to highlight a few of the really important ones. The first one that's important is cohesion and adhesion. This is the stickiness of water. So water is sticky. It sticks to itself and it sticks to other things, and the reason is because of the hydrogen bond. Just a note, all of the properties of water are due to the hydrogen bond. It really is a unique aspect of water, and it gives it these really neat properties. So cohesion and adhesion, the important biological importance of cohesion and adhesion would be something called transpiration in plants. So in this class, we're going to be studying the photoautotrophic food web. We're going to be looking at solar radiation and how plants convert that to carbs, and then how animals go ahead and consume plants, and so on and so on. Well, plants, in order to do photosynthesis, they have to have water. It's actually an essential molecule, an essential input for photosynthesis. And the interesting thing is that plants get water out to the photosynthetic surfaces in the leaf by water moving up through the roots, up and out to the leaf. So water defies gravity. It moves against gravity, and it's able to do this because of actually two properties of water, but the first one is cohesion and adhesion. Water is sticky. So cohesion and adhesion, stickiness towards itself and other things. So here's how this works. Inside of a stem or a trunk of a plant, what you have are these tubes. And these tubes are what water and nutrients move up through. So water, when you water your plants, you water at the roots, water deeply at the roots, right? And water will move up, and it will actually move through these straws all the way up and then out to the leaves. At the tips of the leaves, actually the undersides of the leaves, you have these doors that open and close. And when these doors open, water evaporates. And that's the driving force of transpiration, actually, is evaporation from the leaf. Because when water evaporates from the leaf, what you have is basically a vacuum. It's like sucking on the end of a straw. And when water evaporates, that moves more water up the tubes. And so water is continuously coming out to those leaves that are trying to photosynthesize. Now, where does cohesion and adhesion come into play? Those come into play because water sticks to itself. Even though it's breaking every few trillionth of a second, the collective hydrogen bond holds it together. And so you get a steady stream of water in those tubes. Right? You don't get pieces of water. You have a steady stream of water. Adhesion comes into play because water actually sticks to the tubes as well, so that as water's moving up, it doesn't fall right back down. It stays in that straw, and then evaporation happens, and it moves up, and it moves up, and water molecules get out into the photosynthetic surface. The second property of water I'd like to talk about is specific heat. Specific heat, for water, water has a really high specific heat. And what that is it's the amount of energy you put into a system to raise the temperature or lower the temperature by 1 degree Celsius. So when I say that water has a really high specific heat, I'm saying that it resists temperature change. And that has tremendous biological importance on Earth because it means that our large bodies of water can store heat. So when you think about climate, Okay, so not weather changes, but long-term weather patterns, climate, and the distribution of heat on our planet. Water plays a tremendous role, if not the primary role in that. So you have sheets of water in a large body of water at different temperatures and thus different depths, and they're moving, and when they butt up against land masses, they distribute their heat. Bodies of water, though, they are slow to give up heat. We're going to talk about evaporative cooling in just a minute, but it's related to that. And so these large bodies of water store tremendous amounts of heat for us. If you live in a city that is near a large body of water, your daytime highs are lower and your nighttime lows are higher, so you don't get as much temperature fluctuation because that body of water is buffering that land mass. So you can stretch that out to a global scale and think about the hospitableness of our planet and the role that water plays, and it's because of specific heat. The third one is surface tension. Water has a very high degree of surface tension. That's important, of course, because it provides environments for other organisms, but it's tremendously important when it's paired with specific heat because it inhibits evaporation. It encourages large bodies of water to hold their heat. Okay? So this is how it works. If you have a beaker of water, so like this beaker right here, this is a drawing of that. So this beaker, the water molecules that are in the middle, and they're not at the air-water interface, like that guy is. So this water molecule in the middle, it's being pulled through hydrogen bonding, breaking and forming all around, right? All different ways. But water molecules at the top, there's no pull at the top to pull them up. So what happens is this water molecule gets pulled down and out, and it forms a tension on the surface of the water, and that's surface tension. And so what that does is that inhibits the release of water molecules moving really fast from evaporative cooling. And so that helps maintain the energy in the system. It keeps the heat in large bodies of water. This is my son Grayson at age 4 months, and this is a good image, I think, because it displays some of the properties of water that we just discussed. So couple of things. You have cohesion and adhesion, water sticking to itself, forming a steady stream that we talked about the importance in transpiration. You have adhesion to the lip, and you have surface tension that is creating a ball at the bottom. It's a little bit elongated because of gravity, but all the molecules of water at the air-water interface are being pulled in, and so you get a ball. All right, so that concludes our section on water. And now I'm going to introduce you to the organic polymers. This is your common misconception for organic polymers. The first is that the function of DNA is just for reproduction. When we looked at the characteristics of life in your Introduction to Biology lecture, we said that DNA is used for reproduction. What else is it used for? Fiber is not a carbohydrate. Oftentimes people think fiber is in its own category. So stay tuned to see what the answer is to that. All fat molecules look the same. A fat is a fat. Fats look the same and fats behave the same inside of our bodies. Protein in the diet is used for energy in the body. So watch the video and see if you can discount these misconceptions. The organic polymers we're going to be looking at, boy, we will be working with these all quarter long. And there are 4 main types that we're going to be looking at. The first polymer is the nucleic acid. So nucleic acid, an example would be something like DNA. The second is proteins. Proteins are going to be incredibly important for us. Living things are basically made up of protein and water. And so our discussion of protein, it's paramount to understanding living things. And so we will be doing protein. We will also be looking at fats. Another word for that is lipid. And we're going to be looking at two different types of lipids. We're going to be looking at the type that we eat, the triglyceride, and we're also going to be looking at the lipid involved in cell membranes, the phospholipid. The last one we're going to look at are carbohydrates, and those are our energy molecule. Okay, so that's an overview of where we're going. We're going to work through each one of those. The first thing we want to talk about before we get started into the specifics of this is we want to understand the difference between a polymer and a monomer. A polymer is just a really big molecule composed of repeating units. You can think of these as Lego bricks. And when the Lego tower is assembled, that's the polymer. You can see that here. These are polymers. And they have these repeating units that are nearly identical. Each of them will be slightly different. So this was a protein, these would be different amino acids. This was a carbohydrate, these would be various monosaccharides. So the repeating units change a little bit, but they're very similar in structure. The monomers are the individual Lego pieces. And you can see those up here. So these would be individual amino acids or individual monosaccharides. Okay, so why is this important? This is actually a really neat design of living things. If we think about this in humans, if this is a protein that I'm eating, so let's say that this is edamame that has protein in it, and I'm eating that as a part of my dinner. That's going to go through the gut, but it's too big to take across the gut as a whole protein. So what you're going to have to do is you have enzymes and water that come in and break up these bonds. And the monomer, or the essential amino acid, is what's carried across the gut. So each of these individually are carried across the gut, and those move around in the bloodstream. And then cells will draw them in and put them together in a new order that's functional for its own cell. So they'll create a new protein that's something that they want. So it could be something like a membrane transport protein or a ribosome. So the functionality of this is tremendous, where you take things apart to get them across the gut, but then you can reassemble them in new ways once you get them into the blood and into the cells. Carbohydrates are the first polymer that we're going to look at. Carbohydrates basically are sugars. You have simple sugars and you have complex sugars. You've heard of both of these. You read about sugars on food labels. You hear about things like added sugars. We're going to get a chance to talk about those when we do nutrition in class. But essentially what you see for complex carbohydrates, the monomer is a monosaccharide. It's one of these rings. So one of these rings, when it's bonded to others, it creates a big, long chain. And those are complex carbohydrates, and they're what you find in vegetables. They're what you find in parts of fruits. They're used in things like cell walls in the vegetables. We're going to be talking about fiber, which are basically these long-chain carbohydrates. We're going to talk about soluble and insoluble fiber. Insoluble fiber is not going to be able to be cut up and surrounded by water and taken across the gut. Soluble fiber is, and there's going to be a big difference between those two and the functionality that they have on human health. The second that we want to talk about is our proteins. So proteins, you're going to get a really basic introduction right now, and then we're going to revisit proteins a little later in the class for exam 2. So proteins are made up of strings of amino acids. So the amino acid is the monomer, and the protein is the polymer. The function of proteins are far-reaching. If we are mostly protein, then obviously proteins perform really diverse functions. They're going to do things like whole system movements. They're going to be involved in making other proteins. They're going to be involved in immune function. They're going to be involved in cellular transportation. They're going to perform many, many, many different functions. The one thing they don't do, though, is provide energy. They are not an energy molecule. And that is so strange to think about because we consume protein in our diet, and I think a lot of Americans think of this as energy, an energy source, and it's not an energy source. Your carbohydrates are your immediate energy source. Fats are an energy source. Proteins are not an energy source. We eat protein to make protein. Okay. So this is a list of all the various amino acids. These amino acids vary. You can see what varies on them is these groups down here. Everything else is the same in terms of the chemical composition of them. These groups that hang off the ends, though, these are called R groups or reactive groups. And they're really central to understanding protein formation. So once you eat protein, goes across the gut into the blood, cells pull them in. So cells are pulling these individual ones in. And when those cells make proteins, the R groups are really important to how those R groups, how the protein looks. So the R groups determine the shape of the protein. And in biology, form is function. So what you look like is what you act like. All right, so I'm going to give you an overview of protein structure, and then I'm going to move on to the document camera, and I'm going to walk you through how a protein is made inside of a cell. So this is a protein structure overview, and this is a good slide looking at the various levels of protein structure. It gives you an idea of complexity. Proteins are so very complex. What would be good, though, is to talk about what comes before A. This primary structure that you see here, you've got all your amino acids in a row, okay? Basically, you're creating the large Lego, right? So you've got all your different amino acids in a row. But how does a cell know to put those particular amino acids in that order? How the cell knows is because the job of DNA in the nucleus is to code for the order of amino acids. So when you talk about an individual's genetics or genes or traits that they have, what you're really talking about is protein synthesis. Your cells, most of them, are mini protein factories. And if they need to make proteins to survive and DNA is running the show, then DNA in the nucleus of eukaryotic cells is determining what those proteins are going to look like. So DNA is running the show here, and it's determining the order of amino acids. And that order is really important because it means you have different R groups in a various order. And as I mentioned earlier, the order of the R groups determines the shape, and the shape determines the function. So what we have here for the primary structure of a protein, what we're going to call this is a polypeptide chain. And it's like a string of beads. And you have all these various shapes in a particular order, and they're all in a long strand. And that's the primary stage of a protein. And it's probably the most important. Because if you don't get the order right, it's not going to look right in the end. So it's really important not to have mutations in DNA so that you don't end up with yellow, yellow, yellow instead of green, yellow, yellow. Because that yellow is going to carry a different R group than that green has. What happens in the secondary stage of a protein is that string of beads, the R groups that are hanging off of it, some are getting attracted to one another, some are getting repelled from one another, and they start folding and twisting into shapes. They move into shapes such as the alpha helix, and you can think of a slinky. So you get that alpha helix shape in part of the protein. In other parts of the protein, you get this beta pleated sheet, kind of like an Asian fan. So what we're going to call the secondary structure of a protein is alpha helix and beta sheet. So we have polypeptide chain, we have alpha helix and beta sheet, but you're actually not done yet. This is how complex proteins are. You're going to take that complex secondary structure, and for tertiary structure, you're going to fold it again. You're going to fold it in on itself. And what you can see here, this tertiary structure, it's very complex, and you have alpha helices, and you have beta sheets, and you have straight regions. There's a diversity of folding that's even within that structure. This tertiary structure we're going to call globular protein, and that is where most proteins are functional. And what I mean by that is it's ready to go out and do its job. So if it's a membrane protein, it's ready to go ahead and embed itself in the membrane and start moving salts in and out, or whatever its particular job is going to be. Some proteins move to the quaternary stage, but not all of them. Red blood cells are an example of where you have 4 of these globular proteins connected together. Ribosomes are another example where you have multiple proteins connected together. But most proteins are functional at the tertiary stage. Okay, I'm going to move to the document camera so that we can see how this works within a cell. Okay, I'm going to draw a cell. And inside the cell are organelles that you've been reading about. And the first that we want to talk about is the nucleus. Inside of that nucleus is DNA. And so we'll draw DNA. So the function of DNA is to have these genes, these gene areas. So we get a sequence of DNA, and we talked in an earlier lecture about these nucleotides, right? So you've got A, T, G, C, let's say, part of the gene, and that gene is going to code for the polypeptide chain. But DNA in eukaryotic cells is conserved. And so it would be very reckless for a eukaryotic cell to send the whole copy of DNA out of the nucleus, out of its protective covering. And so what happens here is a copy of DNA is made in order to conserve the code so that no errors occur to the master copy. It's better for errors to occur to a disposable copy. And that disposable copy is called RNA. So a copy is made, and then RNA is actually moved out of the nucleus. I'm going to draw the endoplasmic reticulum next. I'm going to abbreviate that as ER. And on the ER, there are ribosomes. This is where the RNA goes. We'll talk about different types of RNA and how all this happens a little bit later in the course. For now, we're just doing an overview. So that RNA slips out, and it continues to move over to the ribosome. It moves through the ribosome, and the ribosome provides the enzymatic action to link amino acids. So amino acids, remember, that came from protein in your diet, and so you have amino acids in the blood outside of the cell that get moved into the cell. And then those are, they're just kind of floating around within the cell. So you've got different types of amino acids inside the cell, and those get facilitated through bonding patterns to create a polypeptide chain, the correct order of amino acids. Okay, then that chain, that primary structure for protein, the polypeptide chain that's made on the ribosome, slips into the ER. The ER is like a whoopee cushion that's been folded up and down and up and down, and inside of those pockets are enzymes. And so this guy slips into these pockets and it gets folded into all the various stages. So alpha helix, beta sheet, and globular protein. So you get the secondary and the tertiary stage of a protein. So all of the folding happens in the ER. The function of the ER is to fold polypeptide chains. And then that leaves, and it goes somewhere called the Golgi apparatus, which is another membrane-bound organelle. All these organelles work together in a system called the endomembrane system. You have the nucleus, the ER, and the Golgi. And so that protein goes over to the Golgi apparatus. And what happens to it here is it gets flagged. Proteins get flagged so that they can tell other proteins and the cell what their job is in the cell. And if we're making a membrane transport protein, then this guy, once it gets flagged, is going to go out to the outer membrane, and it's going to be embedded in that membrane so that it can do things like move salts or bring in carbohydrates. Okay, so that's the endomembrane system. One of the things that you want to really avoid with proteins is something called denaturation, and that's when proteins unravel. The unraveling of proteins is really dangerous because if form is function, a loss of the form can lead to a loss of function. So, for instance, when you get sick, if you have a virus that you breathe in through your nose and it goes into one of your cells, what it's going to do is, like we talked about before, hijack your DNA and get itself to divide. What your body's response is going to be is to give you a temperature, right? Raise the body heat inside of your body. And so what happens is the molecules, everything starts shaking more. Temperature is a measure of molecular motion. So you increase the temperature, you increase the motion. And the idea is that, well, let's shake things so much that proteins denature in the virus and the bacteria. And so temperature, having a temperature is something that can help degrade proteins in enemies that are inside of you. You know this to be true in terms of proteins in eggs, for instance. They firm up, they literally look different, they feel different once they've been heated. Those proteins are literally changing shape. Okay, so the other thing you'd want to know about proteins is that, you know, what happens when you get the sequence wrong? What happens when you do have a mutation that's not good, that's deleterious, and it actually leads to disease? So the connection between your proteins and disease is an intimate one. It's one in which changes made to DNA can lead to disease. And so in an example of that, this is an example of sickle cell disease. You may have heard it as sickle cell anemia. And what happens is there's a letter change, a nucleotide change in the DNA. And it leads to only one amino acid chain in the whole entire chain. And that causes structural and functional differences. So a normal, happy, healthy red blood cell is this donut shape, and it's about 10 microns, 7 to 10 microns in diameter. Your smallest blood vessel is about 1 micron in diameter, your blood capillaries. So these are huge compared. They're 7 to 10 times larger than your smallest blood vessel. And that is one of the beauties of the design, right? Is because these large red blood cells literally squeeze through the blood capillaries, and that aids in gas exchange. It facilitates diffusion, basically, the movement of CO2 out into the blood and oxygen from the blood and into the cells. When your blood cells look like this, though, you're not as effective at carrying gases, either away or to tissues, and you're more likely to get them stuck and clogged in blood vessels. So let me show you how that works into the diagram we were drawing. So what I'm saying is that there's a change at the DNA level for sickle cell disease, where you have a mutation and you have a new nucleotide that's put in its place. That changes the RNA. You're going to have a different nucleotide in the RNA. And when that RNA is fed through the ribosome, it means that you're going to have a change in this chain. So instead of this first one being a triangle, let's make it a circle. And it shouldn't be a circle, it should be a triangle. And what happens here then is this shape change is off. When the shape is being formed in the ER, because you have a new R group there, that shape is different. Then when it goes to the Golgi, it gets flagged to do its job, but it can't do its job. Can't do its job as well as those normal, healthy red blood cells. Okay, so that's a little bit about mutation. We're going to be doing a lot more on mutation in class. Okay, this next section we'd like to look at is our lipids. Lipids are fats. There's two types, like I mentioned earlier. We're going to be looking at phospholipids, which make up cell membranes, and we're going to be looking at triglycerides, which are the fats that we eat. Let's look at phospholipids first. Phospholipids are really interesting molecules. They have a polar head and nonpolar tails. So their polarity is split. The heads really like water, and that's because you can see there's a plus sign right there, there's a minus sign right there, so they have some full-blown charges in that head. And so water likes the head, but not the tails. It's all CH2s. And so we can draw it like this, where we've got the head in the water, and then you've got the fatty acid tails sticking down. When you put phospholipids in water, what happens is those tails naturally shy away from water, and they move towards one another. And so they make a lipid bilayer with the head sticking out in the water and the tails forming this hydrophobic domain. This is fantastic because it allows for the movement of some molecules in and others not. So we'll be talking about different molecules and ions and how they move through the membrane, because this membrane is going to be a significant barrier to certain molecules moving in and out. If you have a molecule that's polar, for instance, it's going to be fine with the heads, but it's going to be repelled from those tails. It's going to want to go the other way. The other type of fat we're going to look at is the triglyceride. So I'm going to do a little demo for you before we get into detail of triglycerides, and it's going to involve triglycerides, which is what oil's made up of, and also what butter is made up of, and then it's going to involve a detergent. Okay. Okay, so everybody knows that when you put oil in water, it does not mix, right? So if I go ahead and I pour olive oil into my water, you're going to be able to see that it doesn't mix. And in fact, those hydrophobic molecules like each other, but not water. And so they're forming these large globs because they're shying away from water and they're moving towards each other. And in the end, this will most likely be one huge, large glob. Like those guys are trying to incorporate almost, they have a barrier of water in between them right there, right? So oil and water, things that are hydrophobic, do not like hydrophilic molecules, polar and non-polar. Well, let's do something. You guys know about detergents and soaps because it's the way that we get things like bacon grease off pans. In the absence of something like an abrasive agent that we can scrub it off with, we use soap in the modern world. And what soap is it's kind of like a phospholipid. It has a polar region and a nonpolar region. That's very different than those triglycerides that are completely nonpolar. So let's look and see what happens chemically when I put a little bit of soap in here. So I've added a little bit of soap, and what I'm going to do is I'm going to take a stir rod, and I'm going to provide a little bit of molecular motion, not a lot, just a little bit, so that the phospholipid-like molecules can actually make points of attachment. And what you're seeing happening here is you're seeing the detergent molecules, which are kind of like phospholipids, being able to attach to the fat with their nonpolar side and water molecules with their polar side, so that water can, in fact, form spheres of hydration. So you can see sphere of hydration around this globule, this one, and obviously if I stirred even harder, those get smaller and smaller and smaller. So truly, phospholipids and detergents are able to bridge that polar-nonpolar barrier. Okay, let's talk a bit more about our fats. What we want to understand in terms of triglycerides is degree of saturation is going to be an important thing for us. So first of all, with triglycerides, your monomer is the fatty acid tail. So triglycerides are considered polymers. Their monomer is a fatty acid tail. Their function is energy storage. That's different than the carbohydrate, which was immediate energy use. This is energy storage, and the reason is because they are totally hydrophobic. Just like the oil that I put into water, they form those globules. So what a perfect molecule to use for fat storage. So we have our adipose cells, and we have fat globules that we store in those cells. It's really a brilliant way to store. If we had a storage molecule like the carbohydrate, which we do have limited stores for, but if that was our main storage, we would have to be a lot bigger and bulkier because the carbohydrate's like water. And so if you're going to store carbs, you got to store water with it. It's always surrounded by water. And so it's just a lot bigger and bulkier. All right, so you know function, you know monomer. So let's talk a little bit about types of fats. All right, so degree of saturation. When you look on the back of a label and you're reading, okay, 14 grams of saturated fat in oil, 2 grams saturated, 12 grams unsaturated. What does that actually mean? This, by the way, also pertains to phospholipid tails. Phospholipid tails can be bent or straight, saturated or unsaturated. So let's take a look. If you have saturated tails, you're talking about something like butter, where it has the maximum number of hydrogens on each of the carbon atoms. There's no double bonds, and so the chain hangs straight. Now, because the chains are all straight and they're all packed together, you end up with something that is solid at room temperature, like butter. And that's going to perform a bit differently when we talk about nutrition than something that has a kink in it or a double bond. An unsaturated fat means that it's not maximally saturated with hydrogens. It's missing 2 hydrogens. So it has a double bond in it, and that forms a kink in the chain. You've got this kink here, and that spreads the triglycerides out, or the phospholipids out. In this case, triglycerides in oil is liquid at room temperature. So we have saturated and we have unsaturated forms of fats. And there will be some implications because of that. Okay, last thing we're going to talk about are nucleic acids. Nucleic acids examples are DNA and RNA. Okay, so these are information storage molecules. They are built of nucleotides. So here's your nucleotide. It's made of a phosphate group, which has these negative groups on it, so it's going to like water. It's got a sugar, this is definitely going to like water, and it's going to have a nitrogenous base, one of these four. If you're talking about DNA, it could have U if you're talking about RNA. I'm not going to worry about that for now, though. What we want to know is that nucleotide is the monomer of a nucleic acid, and when they stack together, they form these long strands. DNA is double-stranded, so you've got basically nucleotides on this side and nucleotides on this side, and then the bases interacting in the middle. For RNA, you're just going to have one side. It's a straight chain. But function, information storage, and we're going to do a lot more on that in class. One more thing I'd like to mention on the nucleic acid area is ATP. So ATP, adenosine triphosphate, is cellular energy. It's not an information storage molecule, but look at it. It looks like a nucleotide, right? And that's because it is a nucleotide. So it has a sugar, nitrogenous base. Adenine is the nitrogenous base. DNA has adenine too. And then you have phosphate, but you have 3 phosphates. And this never stacks. So the reason that DNA and RNA are information storage, the reason they can store is because they stack into these long chain polymers. ATP never forms a polymer. It is a nucleotide. End of story. Okay, and so this is a bit of an exception to the rule for nucleotides. So that concludes the discussion about chemistry, water, and organic polymers.